Everyone, this is Yushi and welcome to my lab. So for this lab, we are going to talk about the RNA sequencing technologies and how to use all the RNA sequencing classic bioinformatic tools with application in our homework and related assignments. So I hope you guys could get familiar with all these tools and get hands-on skills about how to explore some really new and original bioinformatic tools with the application in every area. So uh, you can find all my lab material on the GitHub. I believe I have linked all these files in the Canvas site. And okay, now let's begin and go back to our slides. So some outline. First of all, we would touch some RNA sequencing analysis workflow, and then talk about the RNA sequencing tools. And since 70% of you guys haven't touched Odyssey issues before, I would hold a special Odyssey Fest for you guys. And the last session is about going through all the assignments one by one. So to begin with, here's our announcement. Please do not share your code with fellow students via GitHub, and please make sure your repo is private if you have to use the GitHub to share your code. And please also double check you have access to Odyssey, and the folder where we store all our data. You might need it for homework 3, 4, and 5. Well, I would go through all these homeworks with you for the remaining 6 weeks, so please don't get bored with me. And please shoot me an email if you are not blessed with access to that. The last tip is that please start early on homework 3 because we have to run multiple jobs on Odyssey for homework 3, especially for question 1, and you guys might get stuck at that step if you have to wait for a long time. So good luck for this part, and start it early would never hurt anyone. So the basic idea about the RNA sequencing, here is a concept about that. You can check the 2009 Nature paper about that. It's a really long paper, but really good. And basically, it compares the alignment to the library that they have built. So when you try to do the RNA sequencing alignment, basically you might need 2 FASTQ files or some similar files to do that. You can check the details from the original paper. I wouldn't touch that for this lab because I think you guys have already get familiar with the concept in the lecture of Shirley's lecture. And so RNA sequencing is actually doing some fragment alignments. You guys have already touched star in the last session of our lab. I guess it's lab 4. So star, how's that star going? I bet it consumes a lot of time, right? Typically around 1 hour or so. So start slow since it's doing the alignment to compare all the fragments to some really large transcriptome or the whole transcriptome. But for this lab, we would recommend introduce you guys another new bioinformatics tools that was built around 2017. So it's called Salmon, a nice name. So Salmon is fast because it adopted some pseudo-alignment concepts of rapidly alignment rates. The reads for Salmon are classified according to their fragment equivalence class. It could optionally remove sequencing biases and compute coverage via likelihood maximization. It adopted the similar idea and concepts and results to near optimal probabilistic RNA sequencing quantification. If you are interested, you can just check this 2016 Nature paper in biotechnology. It talked about all the basic ideas. So since Salmon adopted some bootstrap algorithm, it's faster than Star, but it decided on the reliability of the Salmon results might vary. It depends on the number of bootstraps that you actually use. If you use the right number of bootstrap, you would get the reliable results. We will touch that later when we talk about the details about how to run Salmon. So here's just a review about how to run star. Star needs to be run on some cluster computing. For us, we would use the Odyssey cluster computing. To use the Odyssey computing, you need to use some bash code, also begin with, we call it as shebang. So there is a hint about how to use that. Let's go through them one by one. The first line is can't be changed, just the title of the shebang. And The special code, the dash capital N denotes the number of nodes that you want to use. The dash N denotes the number of cores that you want to use in this nodes, in this specific nodes. And the dash T is the time that you expected your job to run. Well, actually, this is the upper limit of the time. So it could be as large as around 7 days. So you cannot exceed the 7 days. And the dash P is the partition that you want to use for Odyssey. So for Odyssey, we got many partitions. I will give you some hints about how to improve your speed by making full use of all the partitions on Odyssey. We'll talk about that later in the Odyssey first session. And bash memory is the total memory that you expected to apply for this job. So dash O and dash E would store the output and the arrow files. And if you use the dash dash mail command here, you can send the result to your email. And once your job has been finished, you would get an email. That's easy. Okay, now let's begin our salmon. It's all about how to train your salmon without fish. So salmon is a fast, accurate, and bias-aware tool for quantifying the expression of transcripts using RNA sequencing data. Well, it's called the first transcriptome wide quantifier to correct for fragment GC content bias. It's fast because it adopted some pseudo-alignment techniques. It's accurate because it combines a new dual-phase parallel inference algorithm and feature-rich bias models with an ultra-fast read mapping. It's bias-aware because it could account for sequencing-specific fragment GC and positional biases. Well, this is called GC bias. There's a whole Nature paper about that. If you are interested in that, shoot me an email and I should send you where to find that specific paper. So the first step to use the Salmon is to build a Salmon index file. That's very beginning for your question 1 in the first part of your homework 3. So in order to quantify transcript level abundances, Salmon requires a target transcriptome. This transcriptome is given to Salmon in the form of a multi-FASTA file with each entry providing the sequence of a transcript. I have already prepared this transcriptome, which was stored on Odyssey, and I have pointed out the paths in your homework. So next, we are supposed to build an index based on our transcriptome. My transcriptome is based on the HG38 human genome. So when you are trying to do some downstream analysis, remember to set the threshold or the indicators in coordinates with HG38 version instead of HG19. These two varies a lot. Just pay attention to that. So this index is a structure that Salmon might use to quasi-map RNA sequencing rates during quantification step. The index needs only to be constructed once per transcriptome and can then be reused to quantify many experiments. I bet for question 1 you have to build the only one index while you have to run multiple alignments. I think there are 4 alignment jobs that you need to run. So here's an important note. Please do not build the index on the genome of the organism whose transcripts you want to quantify. Well, this is like you are doing some comparison and doing some annotations according to exactly the same experiment example. It's we cannot imagine the result. So to build the Salmon index, it's very simple command, only with 2 instructions. So basically go with Salmon index. Well, dash T followed by the transcriptome file where you stored the transcriptome file, FASTA file. Dash I indicates the location where you want to store the output. So this is basically the name of the folder that you want to build the Salmon index. By default, Salmon adopted some quasi mapping method, but it also applies some SMEM-based lightweight alignment method. Well, the first one is more reliable, basically, but it consumes a lot of time. But quasi mapping method is really fast, and it could be as reliable as the first one if you can choose some reliable threshold. I would recommend you to leave these things out, and just keep the first part, and let the tool to select the algorithm. Just the default setting is highly recommended. This is a example code that could help you to build the index. So to begin with, combine all these commands into an .sh file and dispatch this .sh file on Odyssey. The -d followed by the location where we store the transcriptome. Well, here's the location that we store the transcriptome for last year. For this year, I changed it. You can check my updated codes in slide 32 or so. And -i is the location where we want to store the output. Well, this is the name of the folder. And here, just give you some hints about what the output look like. So remember, I saved all this Salmon index folder under the temp location, and within this folder, you can find all these files, and all of them are needed for the quantifying step. So the second step is about Zabo quantifying. For this step, you need two paired FASTQ file for each alignment. And the instructions are also very simple. Basically, you only need 4 instructions. So -i indicates the index file where you store the Salmon index, which is the output of the step 1. Remember that. And -il is the library that we want to use for this alignment. Capital A means automatically, so we let the software to decide which library to look for automatically. And -1 and -2 refers to the FASTQ file that you want to align together. The order of these two files doesn't matter too much. You can switch them. -o is the output of the quantifying results that you want to store. Well, I store them at the same location as the temp file. And as I mentioned, Salmon use some bootstrap methods, so the number of bootstrap really matters. Well, I would recommend you set it as 100 or so. If you set it larger, no doubt you would get some more reliable results, but it might be time consuming. And so 100 is highly recommended. And -p is the threshold that Salmon would use for running this job. It shouldn't exceed the number you applied for the whole job. And -gc bias would correct for fragment level GC biases in input data. This is actually the advantage of Salmon tools. So do remember to include this code into your script. And after writing all this, you have to write all these commands into one .sh file, and then submit this .sh file to your cluster to your cluster's home address, and then dispatch this this .sh file to cluster computing. So here's a brief idea of what the quantifying result look like. Basically, it's a txt structure file ended with the .sf. You can find the TPM transcripts per minute for each transcript. You can also get the gene begin with the ENSG of the IDs. They are paired with each other one by one. I will show you how to find their specific matches between the transcripts and the gene ID in the R session. And I bet there's a question asking you to pick out the gene with the highest TPM. So you can do some data manipulation based on this quant.sf file in your R and try to pick out the gene with the highest TPM. So this is difficult, not the end. We need some downstream analysis. What we use for this lab is the DSEQ2, and there are other sources that you could use for downstream analysis. I posted all the links here, and feel free to check them out. So here's the end of the Salmon tutorial, and do enjoy your Salmon. So, to give you some more hints about the first question, I bet this is what you need for the first question. So to combine them into a .sh file, and for this step is building the Salmon index, again, -t followed by the transcriptome, which you want to use for building your index. Dash I is the output of your Salmon index. So here, as I mentioned, is the location where I store this human HG38 version of FASTQ files for transcriptome. It's very large, but you can find it on Odyssey. Do not copy it to your home address, just use it. Okay, after you have built the index, remember the location where you store the index, and you have to put the output of the first step into the second step as the -i command. This is the location where you store the Salmon index, and I have already explained all these steps. Then you have to combine them into a .sh file and dispatch the .sh file to the cluster for computing. Oh, by the way, do remember to change your email address. Do not send it to me. I got a bunch of emails, so yeah. Then you have to run your own alignments. Use the commands given earlier to run your own alignments in homework 3. Just remember to make full use of all possible partitions on the Odyssey. I'll touch that in the Odyssey first. And do manage your files. Please construct a file to your local folder, batch submit your job to the cluster, outputting files to your assigned local folder. Upon completion, you may receive an email. You can also view the snarm files and the log files by using some basic operation commands. Well, the abundance estimates are given in the .sf file. You can copy this to your local directory, to your PC, and input them into the R for downstream analysis. So this is the following part of the downstream analysis. For this lab, we would talk more about how to use DSEC2 packages for the downstream analysis. These are all necessary packages that you might need it. So just to check whether you have already got them in your R session. If not, if you have already got them, just jump these steps. So one thing you can verify, for this lab, I adopted some data for the mouse experiment. So I might use this transcriptome version for references to do the annotations of all the genes and transcripts. But for your homework, you might need the version for human because that's experiment based on the human tissues. So just remember to change the reference for doing the transcripts and the gene annotation by loading another package. You can also change the version to the most updated one. I guess for now the version 86 is the most updated one. Okay, for this mouse experiment, basically I have a group of control and a group of treated. I got 3 replicates for the treated samples and 3 replicates for the control samples. So to give you a hint about how this data look like, I store them all in data. So you can see that these three comes from the 3 replicates for the treated sample, and these 3 comes from the alignment result of the control samples. Well, they are all downloaded from the cluster. They are all outputs of the Salmon alignment, and you can also download all these files you have to download them from the cluster and into your own PC, into some specific files such as I named as data. And yes, for this command, I just read in all the files ended with SF under the folder in data, and read them as a example table. Okay, so for this step, we want to do some annotations for both the transcripts and the genome. So this is the mouse version that we used. You might need to change them to the human version for your homework. Well, one thing to mention, this is for lab session, but for homework, for the lab, we got 3 replicates for treated and 3 replicates for control. But for the homework, we got 2 replicates for treated and 2 replicates for control. So you might change this number 3 into 2 and change this number 3 also into 2. So the name you might also only need 2 replicates and 2 control. So you have to do some adjustments for the code. Okay, then we do the D-seq analysis. Before that, we might do some QC steps, such as remove all rows with a total row count less than or equal to 1. This is basically what this step is doing. And this step might consume some time, and then we run the D-seq analysis. After that, we can got the log2 for change. So when the log2 for change is larger than 0, we consider it as the upregulated genes. We store it as upr. And when it is less than 0, we consider it as downstream genes. We call it as downr. So we store them in, you can type all the gene names when you try to explore these objects. And you could also print out the number of upregulated genes here and also the number of downregulated genes here. You can also write out the upr and downr for all the gene ID for the following analysis, such as the gene ontology, and submitted them to software such as the David for the functional annotation. To visualize the results, basically we have two ideas to run that. One is the MA plot, and another is the heat map. But here, I would recommend you guys to use the MA plot because the MA plot could help you to visualize the differential expression with some more reliable quantifying result. Well, because the heat map is basically some unsupervised method, it didn't give you some quantifying result. But for the MA plot, you might get which gene or transcripts are differentially expressed between the treated samples and the control samples. Well, the color of the nodes are quite different because the red nodes are actually identified in this experiment. Okay, now is the funny part. It's the Odyssey first. So according to our survey before, around 70% of you guys are not quite familiar with Odyssey. Maybe you guys are familiar with other cluster computing. So no worries, it's quite easy to learn. And this is about how to use how to make full use of all partitions. Just change this single line to make full use all partitions and combine them into one sentence together. So Odyssey would try to choose whatever partition is available at that time for you. If you just choose one partition, you have to stay in this partition until the nodes in the partition are available for you. Actually, I want to give you guys a hint about how to check the information about these partitions. Just type in fast Odyssey. Oh, sorry. Yeah, all the fast guys have a very good summary about all the partitions they have. Yeah, here we go. So you can see that these are all the partitions that fast have. So basically, you can consider partition as the cluster of some nodes that remotely from your login Odyssey. So what we use the most is shared, general, and serial queue. They have various numbers of nodes and cores. Well, serial queue have the fastest speed, but when you are running jobs on serial queue, they might be interrupted if other account with higher priority apply to run the job at the same time. So your job have the risk to be interrupted when you are trying to use a partition of serial queue. But for general, when you submitted your job to partition for nodes within the partition of general, it wouldn't be interrupted no matter how higher priority the other account have. So no worries about interruptions when you are going to run general, but it's slower than serial queue in general. And Sherry, I bet is the oldest version of the partitions that Odyssey have. So sometimes it might give you some errors. Well, these three are highly recommended for uses for complete your homework. Also, you might got your own labs notes. So just remember to check all the indicators that you can use as the time limited, and these are necessary indicators in your Shibang file and the memories that you could use and you could apply for each job. Okay. Now let's get back. Okay, here is a basic operation reminder. I created a small cheat sheet for you guys about some basic bash code. This is about how to get access to the cluster, and you should SSH your account ID, type in your password and the verification code. Then here we are. And when you access the Odyssey, do remember to run job interactively. You might need this command for running your jobs. This is just a switch you to some remote nodes. So if you run the job in your login nodes, such as the holy login 03, there is a chance that you might break down these nodes. So everyone is accessing the nodes, and once you made the error, nobody else could use the nodes anymore. It's a tragedy. So do remember to run jobs interactively. It could send you from the login nodes to some nodes remotely, such as the C19314. And if you, even though you done something wrong on this node, that wouldn't influence other users too seriously. Then some basic commands: ls to view all the files, cd to change the path, and make direction to make new folder, and mv to change the name of the file. So SCP is about how to upload script to the Odyssey. Basically, you can use pwd to get your home address where you want to store the file on Odyssey, and SCP is a local file into the target path. Do remember to do all this in your own PC, not on Odyssey, not on Odyssey. Pay attention to this location. It's all on your own PC, or you might fail to upload your files. And you can also view all these files on Odyssey. So here is what I do for the index job. Here's what I do, the first example about conducting the alignment. So to submit your job to the cluster, you basically need the dispatch code, and you can also use the skew command to monitor all your jobs, what you have been running. If you want to kill a job, well, sometimes you might realize some error before the completion of your current job. You might cancel with the job ID. It would kill the job. Once you finish the job, you can change to the output directory where you store the output and locate the quant.sf file. This is what you need to download to your own PC for the following down-seq analysis, such as DSeq2. You can got similarly to the you also need the SAP command. So similar to before, you can use the pwd to check the location where you store the quant.sf file. Type in the location here, and also the local location where you want to store all the data, and download them. Remember to do all these downloading steps in your own PC, not on Odyssey. I can give you a hint about how the data look like. So for the DSeq data, I do 2 controls and 2 mutants. So I named them as control 1, control 2, and mutant 1 and mutant 2. So this is the result. Actually, I can also show you where I store them on Odyssey. Well, another thing I would suggest you guys to do is just create some cheat sheet for you guys. It's really easy. That's basically some basic commands that you might need it for running jobs or data manipulation or doing research or completing all your experiences or assignments. So sh your Odyssey account ID. Oh, sorry. and type in your password and the verification code. It's located here. You can also get it from your phone. And also do remember to run it interactively. Well, for this step, -p also means the partition. It leads you to the test partition. And -t means the time you want to run this interactive job. It varies. So for me, I applied to upper limit of 5 hours. And dash memory is the space that you needed for this interactive job. Basically, you can see that this is a simplified version of the shebang, the .sh file. Okay, now you can see that we change from the boss login nodes to some remote nodes named as the Holy 7C. Okay, we can see all the files we have here. Just change to Stat115. Okay, I store them in work. Okay, so I store all the output as control1, control2, mutant1, and mutant2 because if you can go back to my slide. Yes, for example, I store the first comparison in control1, so you can find it under control1. This is the file that we need as the quant.sf. You have to download it. So this file is exactly this one. And you can find control2, the second quant.sf file. This is this one. Cool. Let's go back. All right. Now let's go through the experiences. Well, it's homework. I call it as experiences. It gives me more things. So for this homework, we will use the RNA sequencing data of the HepG2 and the U2AF1 knockdown and control, each with 2 replicates. So your target, your first your first mission is to check the original study design, the original experiment design, to find which files are control files and which files comes from the treated examples. So to give you a hint what I'm talking about, if you go to the folder where we store all our data, is this homework 3? Yes, to the FASTQ data. Basically, you got 8 files. Forget the sub FASTQ file. They are used for previous year's homework. So for the remaining part, you got 8 FASTQ file. So 2 of them are paired together to do 1 alignment. So you have to run 4 alignments. So you have to run 4 Odyssey jobs for the Salmon alignment. So what you have to do is to find which 2 of them are paired together. How could you do so? You can check these 2 original study designs. So the first one is for the control examples. Second one is for the treated examples. For example, if we go to this side and check the overall design, you can see this is for control groups. And when you roll down, you can got the information here. So you got a number. See that this is number 178 and the number 229. So here it is. Number 178 and the number 229 are paired together, and that's why these 2 files are compared together to make the first control result. And this is not the end. Go back to the original study design. So this is the first replicate for control. And the second replicate for control you can find here. it begins with the number 768 and the number 773. So here it is number 768 and number 773. So these two files are paired together. You would need to use this for creating as the second control. So basically this one is 768 and 773. Actually, they got exactly the same code, but just change the name of the FASTQ file to be to do the alignment for the second replicate in the control group. I think you got my idea. And another pair of comes from the treated group. You can also check the original study design. So this is the modified one, treated one with the gene knockdown. And here is the name of the files 214. You can got 214 here. And another one is 897. So 214 and 897 are paired together. And another replicate is 500 and 708. So 500 and 708 are paired together. Okay, sweet. Let's go back. All these files are stored on this location. It's exactly here. Yeah, this one. Great. So you already completed the first question. So first question to begin with, you have to build the Salmon index. Again, this is what you might use for this one command. This is where you store the output of the index file, combine them into one .sh file, and submit to the cluster computing. Do remember to change the email address and don't send the email to me, please. And followed by this, you have to do the alignment. This is where the output of your first step, and this is what we talked about, the paired FASTQ files. Well, you have to do it for 4 times. For each time, you just change the paired FASTQ files. I have already taught you guys how to find the paired FASTQ files. If you still fail to find them, just come to my office hours. Then we could run the DSEQ2 analysis. All this could be done in R, but I don't want to do it interactively now. You can check it out by your own. For question 3, I give you some recommendations about the geoanalysis tools. Well, the first 3 are not. These 3 are all for how to analyze this specific gene, and the remaining part is about how to conduct the geoanalysis to see whether you have some interesting findings. Actually, definitely the other procedures to conduct the geoanalysis are highly recommended. Yeah, for question 4, it asks you to try to compare the transcript level and the gene level. So by default setting, the Salmon uses the TX out as false. That denotes as the gene level. So if you change the default as true, it's for the transcript level. And if we change the default as false, it gives you the gene level result. You can basically compare the number of upregulated genes and the number of downregulated genes, and you can also compare the geoanalysis results. I think Shirley has already touched the differences between these two default settings in the lectures. If you are not quite familiar what actually differences was expected to be, come to my office hour. Okay, and for question 5, you want to use the abundance instead of the counts. So what I'm doing is just to assign the abundance to the constant column, and the constant column is what is actually used for conducting the DSEQ data. So I didn't change anything else, just replace the count column as the abundance, and it could conduct the analysis based on the counts. Based on the abundance, sorry. All right, this is what I prepared for this lab. and I would hold my office hours for today until 8 p.m. And for Tuesday, I have hold the office hour until 7 p.m. According to your feedback, I might hold an additional office hour on each Friday since next week, because for this week, I have to I didn't find a place, so I have to run this with Janina to find a space to do so. So I will give you some announcement on Canvas when I finally got an idea where to do so. Good luck for your homework, and thank you for coming to my lab.